encore, les vaccins qu'il essaie maintenant d'injecter à tout le monde sur Terre utilisent la technologie d'édition de gènes CRISPR pour modifier l'ADN et la structure du cerveau des humains afin d'éliminer les gènes liés aux croyances religieuses et d'ajouter des gènes pour augmenter la production de dopamine. Cela devrait rendre les gens passifs et heureux, peu importe ce que leur gouvernement pourrait leur faire subir. Jetez un coup d'œil aux deux vidéos suivantes, l'une de moins de 5 minutes et l'autre de 29 minutes. Si vous êtes pressé par le temps, passez aux 10 dernières minutes pour voir exactement comment ils s'y prennent pour arriver
On the
à
left over here, we have individuals who are religious fundamentalists, religious fanatics, and this is the expression, RT-PCR, real-time PCR expression of the VMAT2 gene. Over here, we have individuals. So let me complete. So over here we have individuals who are not particularly fundamentalist, not particularly religious, and you can see there's a much reduced expression of this particular gene, the VMAT2 gene. Another evidence that supports our hypothesis for the development of this approach.
What you see here is by spreading this virus, we're going to eliminate individuals from donning on a bomb vest and going into a market and blowing up the market.
So our hypothesis is that these are fanatical people, that they have overexpression of the VMAT2 gene, and that by vaccinating them against this, we'll eliminate this behavior. So we have some very, very remarkable data in this next slide. Here we have two brain scans. These are fMRIs. These are two different individuals with different levels of expression of VMAT2. On top is an individual who's a religious fanatic, an individual, and we've repeated this numerous times, that has high levels of VMAT2. Now, this individual down here who had low levels of the VMAT2 gene, this individual would self-describe as not particularly religious. In each case, these individuals were read a religious text. This individual lit up the right middle frontal gyrus, shown here, and that's a part of the brain that's associated with theory of mind. It's a part of the brain that has to do with intents and beliefs and desires. In marked contrast, here's an individual who would not particularly self-describe as religious, and when they're read a religious text, what you see is that this part of the brain called the anterior insula lights up. This is a part of the brain that's associated with disgust or displeasure on hearing something.
So you're suggesting I take a CT scan with me when I'm evaluating people to determine whether I put a bullet in their head?
So the data that I'm presenting here supports the concept that we're proposing, and I think that we would not propose to do CT scans or fMRIs on individuals out in the hinterlands of Afghanistan. The virus would immunize against this VMAT2 gene, and that would have the effect that you see here, which is it's essentially to turn a fanatic into a normal person. And we think that will have major effects in the Middle East.
How would you suggest that this is going to be dispersed? The aerosol?
Well, so the present plan and the tests that we've done so far have used respiratory viruses such as flu or rhinoviruses, and we believe that that's a satisfactory way to get the exposure of the largest part of the population. Most of us, of course, have been exposed to both of those viruses, and we're quite confident that this will be a very successful approach.
This is fascinating. What's the name of this proposal?
Yeah, so the name of this project is FunVax, which is the vaccine for religious fundamentalism.
And you have a proposal already?
The proposal has just been submitted, and I think that the data that I have shown you today would support the development of this project, and we think it has.
Usually I go to neuroscience meetings and I have to explain a lot of the tool development aspects of what we're doing, and here that's kind of just appreciated and understood. So that's been really fantastic. So my lab is interested in understanding how gene regulatory mechanisms in the brain help to establish the physiology and function of the nervous system during development, and then in the adult nervous system, how those same mechanisms can be regulated by activity-dependent, experience-dependent processes to achieve things like memory formation, plasticity of the brain, and can be disrupted in disease states. And so Beverly really nicely introduced this concept of genome editing, and I don't really think I have to introduce this at this meeting. What I'd like to talk about today is not using CRISPR-Cas9 as a DNA editing tool for gene regulation, but using these CRISPR-Cas9 approaches, which have recently been described and can be utilized in a number of different ways. So these dead Cas9 approaches take advantage of inactivation of the two nuclease domains in the Cas9 protein. This basically turns this from a DNA editing system into a genomic anchoring system. And this allows targeted genomic recruitment for gene activation, repression, gene localization, and even non-coding RNA localization in the genome. So early studies showed that you can fuse transcriptional activators to these dead Cas9 proteins. In this example, fusion of a VPR, which is a robust transcriptional activator that's really a concatemer of 3 different activators. Recruitment of this VPR protein to a target gene promoter can drive transcription of that gene. Of course, you can fuse repressors to the Cas9 to accomplish repression. You can fuse reporter molecules for localization in the genome. This is an early result from the laboratory of George Church showing that this VPR strategy works very well for a targeted upregulation of a number of different genes, in this case targeting some of the transcription factors that are responsible for neuronal differentiation. And you can see here not only that VPR was very effective as compared to a negative control, but also versus the previous generation of these activator proteins, VP64. And so we were very excited to use this strategy in the lab. And if you're not familiar with George's work, he's giving a presidential lecture later today. But we wanted to, of course, use these in primary neurons and eventually in the intact brain. And so a lot of these early papers have been describing these systems in dividing cell lines, which are easy to transfect, easy to insert transgenes into. And we're using primary neurons, and we're using intact brain, and we're using a rat model system in the lab, which has not traditionally been amenable to gene regulation in this way. And so this introduced a number of challenges to us. How do we get the genes into the cells that we're trying to study? Of course, this relies largely on viral delivery methods to accomplish this, and that can be an inefficient process in neurons, and you start to care about targeting different neurons in different areas of the brain. And so when we decided to start working with this, one of the first questions we had was, well, first we developed this capacity to introduce this via a lentiviral delivery system because we needed the cargo capacity of this delivery system for the adult central nervous system. And we have a modular approach, and so one lentivirus drives the guide RNA, also an M-shared reporter. A second lentivirus drives the DCAS9 VPR transgene, and we needed to select a promoter for this. The original Church paper used a CMV promoter, and we wanted to try to avoid that in neurons because it's known that this is an activity-dependent promoter. It has several Krebs elements, and so it can be regulated itself by activity states in neurons, and we thought that was an obvious kind of caveat for trying to use this in neurons. There's also some evidence that the CMV promoter can be silenced by methylation and can downregulate expression of the transgene. So in HEX cells, we tried a number of different promoters, EF1 alpha, PGK, CAG, and SYN, and targeting the human FOS gene, which is an activity-responsive gene in neurons. It's broadly used to mark neuronal activity. introduced, you're always worried about off-target effects, and so using these transcriptional activation strategies, you're not really looking for edits to the genome, of course, but you're looking for off-target expression of other genes. So we performed RNA sequencing targeting the BDNF1 or the BDNF4 promoter, and I'm just showing the BDNF1 data here. This is kind of a mirrored Manhattan plot, and so on the y-axis we're showing the log two-fold change. So if it's above the white dotted line, it's increase in expression. If it's below that, it's decrease in expression. And this is stratified by chromosome on the x-axis. So what you can see is that when we target BDNF exon 1, BDNF is the most upregulated gene in the genome, which is comforting. We also identified using an off-target prediction software, Cas-Off Finder, the predicted off-target or mismatch, allowing up to 4 mismatches in the guide RNA sequence. And these are mapped here in orange, and what you can see is that there's no consistent regulation of these predicted off-target hits in either direction. And so we think this is selective not only at the level of specific BDNF transcripts, but at the level of the entire genome as well. There are many other genes changed. BDNF is an important molecule, and so when you overexpress BDNF, many genes do change, but we think these other changes are secondary effects of regulating BDNF and not off-target effects. So if we co-regulate, this system is very amenable to multiplexing. If we co-regulate both BDNF1 and BDNF4, which actually commonly occurs in neurons, these variants tend to go up together after neuronal stimulation or plasticity-inducing stimuli. We find that there's an increase in BDNF protein, about a 6-fold increase. And we wanted to use this to ask what these specific transcript variants do in the context of neuronal function. And so we can just grow primary neurons on these multi-electrode arrays. These are just images of this. You can see the M-cherry reporter coming from the guide RNA expression vector. And each one of these black circles in this image is an electrode that can sense and record extracellular electrophysiological activity of neurons grown in the culture. And we can basically just seed neurons on these electrode arrays, transduce with our viruses, and then record from these multi-electrode arrays at successive days after the viral transduction. And you can see kind of what this looks like here. Neurons exhibit these characteristic action potentials, of course, and we can measure those, quantify them, quantify how many overall action potentials occur, the frequency of those action potentials, how many occur in bursts or kind of discrete, rapid succession of action potentials. And doing that for BDNF, what we can see is that compared to our LAC-Z control, which is, of course, a non-targeting gene that's not found in the rat genome, we see increased frequency of neuronal action potentials. This is just time series data. Each one of these rows is a single neuron that we're recording from. Each single tick is a single action potential. And if we quantify this, we can see there's an increased spike frequency resulting from BDNF upregulation, and there's also an increased frequency of burst firing in BDNF1 and 4 upregulated neurons. So we wanted to take this further into the adult central nervous system. We decided to target a region of the brain that we care a lot about in the lab relevant to drug addiction, and this work is funded by the National Institute on Drug Abuse. And so this, the region of the brain highlighted here in green is called the nucleus accumbens. This is the core subregion of the nucleus accumbens, and this is absolutely critical for development of motivated behavior towards drugs of abuse and is altered long-term after exposure to drugs of abuse. So on the left side of the brain, in one animal, we're going to target our LAC-Z control guide RNA with our DCAS9 VPR fusion. On the right side of the brain, we're going to target the FOSB gene using a specific guide RNA for that gene promoter and express the DCAS9 VPR machinery. And so this is what this looks like. This is an actual slice of the brain. This is from the same animal, just targeting a different guide RNA to different sides of the brain. So the M-cherry reporter is expressed from the guide RNA vector, so we So we can also use this system not just for activation, but using the exact same guide RNAs for repression, just swapping out what the effector protein that is fused to the DCAS9 is. And so in this case, we fuse a crab domain, which would be shown here in red, to the DCAS9 instead of VPR, so we can repress FOS using a FOS guide RNA, just as we can activate it on the top panel. So we can really use this system for bidirectional gene regulation. Again, this is all in rat primary neurons. So again, we wanted to take this further and really harness the what I think is the true power of the system, which is that these guide RNAs can be multiplexed and you can regulate not just single genes, as the field has been doing for the past several decades, but entire programs of genes. Genes, especially activity-dependent genes, tend to be regulated together, not one at a time. And so there are whole transcriptional cascades that occur in concert. And so we wanted to use this strategy to try to either replicate or block some of those transcriptional programs. So a common type of experiment that my lab will do is to take cultured rat striatal neurons, and we're interested in dopamine signaling and the transcriptional consequences of dopamine receptor activation in neurons. And so in this case, we added 1 micromolar of dopamine to a cell culture for an hour, and then we isolated the RNA and performed RNA sequencing. And this is the result of that type of experiment. You can see this is a volcano plot showing the log 2-fold change on the x-axis, p-value on the y-axis. So the genes that are, each dot is a gene, and so the genes that are in the upper right quadrant of this graph are genes that are significantly induced by dopamine receptor activation in our culture model. And what we wanted to do was target as many of these as possible for multiplex gene activation or gene repression to really get at what is the function of this program, not any specific gene in this program, but all of the genes together. So we targeted as many as we could. In this case, I'm highlighting 16 that we were able to successfully target. These are the identities of these genes. And we had to adapt our strategy here. So before I told you about our guide RNA was being expressed from a vector that only contained 1 guide RNA sequence. And so Catherine Savell, a talented PhD student in the lab, had to engineer a multiplexed expression array, essentially, that we could use to target all of these genes at once. And she developed this, what we call dopoplex sgRNA array. So this is dopoplex because we're multiplexing the genes that are regulated by dopamine, and inserted guide RNAs for each of these genes that are upregulated or most upregulated by dopamine receptor activation in striatal neurons in culture. This was a lot of work. It seems easy when I put it on the slide, but Catherine had to clone 52 new guide RNAs, insert all of those individually into lentivirus, screen that in a primary neuronal culture system, and then develop this multiplex vector with Golden Gate assembly and insert all of the successful guide RNAs into the multiplex vector. So not easy, and our control here is going to be just a LAC-Z repeat sgRNA array. So we were able to successfully do this. This is the results of dopoplex induction. This is again showing the log 2-fold change. When we target these genes, we upregulate these genes. That's hopefully not too surprising, but this was really technically challenging to do. All of these genes are upregulated. So what are the consequences of upregulating this dopamine-regulated gene program? There are 670 differentially expressed genes downstream of dopoplex induction using RNA-seq. Some of these are increases, some of these are decreases. But when we look for the functional categories that are enriched in those changes, what we find is that there are enrichment of categories involved in synaptic plasticity, ion channel function, etc. So we think these are kind of key. This first wave of genes that are activated by dopamine are really key transcriptional wave to basically induce this second gene program that remodels the function of neurons. So again, we can culture these neurons on multi-electrode arrays. This is showing what I showed you before. And then we can just compare our LAC-Z controls to our dopoplex targeting. We can see receive induction of this dopamine regulated gene program. And this is really exactly what we predicted, because we think these genes will be repeatedly induced in vivo by drugs like cocaine. And we've also performed RNA-seq to show that many of the same genes induced by cocaine are induced by our dopaplex receptor dopamine receptor activation in vitro. So we think this is at least a proof of principle that modulating a large extent of a gene program can induce a functional consequence in vivo, such as increased cocaine sensitization. Where are we going with this? One of the things that we really want to do is get cell type specificity for this approach, and so Jasmine Ravana, an undergraduate student in the lab, designed basically a double inverted open reading frame version of this using two heterotypic LOX sites, inserting these around the DCAS VPR transgene. We add Cree, it flips around, and this is showing our constitutive version that I've been showing you all along, induction of BDNF1, addition of Cree using this DIO transgene allows selective activation of genes only with Cree on board. But we want to get even better temporal control of this process as well. And so Charlie Gersbach and several others have described these light-activated CRISPR effector systems, or LACE systems. These use a light-sensitive protein such as cryptochrome 2 or Cry2, and you can fuse the effector protein, in this case directly to Cry2, and fuse a binding partner, SIBN, to the DCAS9, so that when you shine a blue light, in this case Cry2 is sensitive to blue light, there's a conformational change in Cry2. This recruits the effector directly to the target gene. We've done this in the lab, targeting guide RNAs for a luciferase reporter, and as you can see here, this is an actual image of the 96-well plate that we perform these assays in. Control guide RNAs, light does nothing. Targeting the EGR1 promoter that drives this luciferase, turning the light on really induces luciferase activity in the system. This is a quantification of that effect. This is 6 hours after turning on the light. We do a whole time course experiment, and what we can see is that basically within 30 minutes to an hour of turning the light on, we're getting upregulation of luciferase. Of course, this requires protein to be made, and so this is actually delayed from the transcriptional effects that we're measuring. And we turn the light off and it starts to go back down to baseline. There are a number of issues with this system. One is that cryptochrome 2, this is the Arabidopsis cryptochrome 2, but it can dimerize with mammalian cryptochrome 2 and induce genomic targeting effects. And so we wanted to avoid that as much as possible. And so we developed a new system based on the FKF1 in-love domain. We call this system FLIC. This is FKF1 light-inducible CRISPR construct. There was a long selection process involved in naming of this system, but this was really driven by MD PhD student Corey Duke and an undergraduate student Nick Southern in the lab, and they presented this work in a poster on Monday night. And so this utilizes a different Arabidopsis light-dependent system, which is responsible for flowering in Arabidopsis. And basically, exactly as I showed you with the other system, you can recruit effector to a target gene using this approach. Corey ran through a really tour de force, a bunch of different architectures of this system, changing the position of the in-love domain relative to the DCAS, adding multiple domains, changing position of the interaction partner Giganthiae, adding multiple versions of that. But what you can see here is that this is the most successful version of this is really comparable to these previous LACE iterations, achieving pretty significant upregulation with lower off-target or background light off effects, and you can see that expressed here. Of course, this also works really nicely at endogenous genes, not just at luciferase reporters. So we're excited about this moving forward and getting temporal control of this process. I'd just like to thank everyone in the lab that's been working on this. Catherine Savell and Corey Duke and Nick Southern were the ones that I mentioned most of the work from today, and also our funding sources for this, and I'd be happy to take your questions.
L'orateur de la première vidéo dit qu'un gène appelé VMA2 a été associé au fondamentalisme religieux. Il recommande de vacciner les gens du Moyen-Orient avec un virus qui supprimera ce gène et éliminera ainsi le fanatisme fondamentaliste. Cependant, comme l'a souligné une source du FSB, un effet secondaire est que toute autre expérience religieuse et croyance en Dieu serait également éliminée. Le FSB indique que le gouvernement américain a déjà mené cette campagne de vaccination au Moyen-Orient. L'orateur de la vidéo plus longue décrit comment ils ont utilisé les vaccins CRISPR à base de gènes modifiés pour modifier la structure du cerveau et le comportement des rats. Il parle de l'utilisation du vaccin pour traiter les problèmes de drogue et autres problèmes de dépendance. Mais là encore, comme le souligne la source du FSB, il explique comment le vaccin peut modifier les gènes pour changer le comportement. Les gens peuvent être heureux tout le temps car leur production de dopamine est stimulée. On peut les empêcher de devenir violents et donc ainsi les rendre obéissants. Alors voilà, la mafia khazarienne veut faire de nous des moutons heureux, totalement déconnectés de leur spiritualité, qui auront été génétiquement programmés pour être obéissants quoi qu'il arrive. C'est pire que ce que George Orwell lui-même a imaginé dans son roman 1984. Voici ce que le MI6 avait à dire sur le sujet. Les vaccins à ARN parrainés par la MK contiennent la technologie d'édition du gène CRISPR, qui n'est pas une inoculation mais de la génomique, et ceci est contraire au droit international et hautement illégal. La soi-disant urgence de santé publique internationale met à rude épreuve les économies mondiales et toutes leurs administrations publiques, y compris la police civile et les tribunaux. Les violations du droit international sont monnaie courante et se multiplient. L'ensemble des données de contre-espionnage que nous recueillons quotidiennement depuis le début de cette soi-disant urgence de santé publique nous donnent de très bonnes raisons de soupçonner que tout ce qui est dit à ce sujet et les décisions qui sont prises par la suite ne sont pas du tout fiables. Nous allons donc mener notre opération de contre-espionnage en concertation avec toutes les communautés de renseignement militaire du monde afin d'aller au fond des choses et d'accélérer la fin de ce qui est si dramatique pour nous tous. Les Canadiens se préparent également à déclarer la guerre aux Rockefeller et à leurs sous-fifres parce que Biden, le lecteur de scénario, a pris des mesures pour protéger les opérations pétrolières de Rockefeller au Moyen-Orient en empêchant les Américains d'acheter du pétrole canadien lequel est non contrôlé par Rockefeller. Les premiers ministres de l'Ontario Doug Ford, de l'Alberta Jason Kenney et du Saskatchewan Scott Moe ont ouvertement déclaré envisager entrer en guerre avec les États-Unis pour l'annulation du pipeline Keystone XL.
The company building the Keystone XL pipeline is laying off 1,000 workers. President Biden has made good on his promise to rescind the permit for the project. And Prime Minister Justin Trudeau is expected to raise it with Biden when the two talk tomorrow. Our chief political correspondent David Aiken joins me. David, is there anything the Prime Minister could say to get the President to change his mind?
Well, no, not really. This was a major commitment of the Biden campaign. They campaigned for months on this, and canceling Keystone is a key symbol of the Biden administration's commitment to fight climate change. Now, Alberta Premier Jason Kenney is pushing to have trade sanctions slapped on the U.S. in retaliation, but trade experts are telling me that not only would that likely be illegal, they're unlikely to work.
I don't think the government of Canada is going to bring an action at this stage in our relationship with the new administration against the United States. I just don't think it's on.
David, what happens next for oil producers in Alberta and Saskatchewan who want to produce more oil and get it to international markets?
Well, as it turns out, exports of Canadian crude to the U.S. has actually been growing even without Keystone. More efficient use of existing pipeline infrastructure. We've seen more oil by rail. But even still, the business case for Keystone has not really existed for years now.
Well, I think what Keystone was and I guess is targeted at would be new projects, new production. And so right now, given the current pricing environment, we don't have any new projects currently under construction.
And let's not forget that the Trans Mountain pipeline, which Canada bought, is being expanded.
Trans Mountain is more than enough extra capacity for the oil we're going to want to export.
Now, Trudeau might want to convince Biden to commit to developing an integrated North American plan that would allow both countries to exploit oil and gas resources and commit to rapidly reducing greenhouse gas emissions. That's a tough order, and there's no guarantee that if they did such a plan, that a Keystone or a successor would even be part of it. Donna?
All right. David Aiken, thanks.
Four years ago, inside the Oval Office, newly elected President Donald Trump brought the Keystone XL pipeline back to life.
TransCanada will finally be allowed to complete this long overdue project with efficiency and with speed.
But now there's a new president in the Oval Office, and Joe Biden has different priorities.
There's no time to waste. Get to work immediately.
One of his first acts was terminating the presidential permit allowing Keystone to cross the border. TC Energy had already waved the white flag, announcing earlier in the day it was suspending work.
This is a gut punch for the Canadian and Alberta economies.
Premier Jason Kenney bet more than $1 billion taxpayer dollars on the project, and legal experts don't see a path to getting it back. The Premier says if Biden doesn't budge, Canada has to fight back.
It is clear that the government of Canada must impose meaningful trade and economic sanctions in response to defend our country's vital economic interests.
There is deep disappointment along the line. Oyen acted as a home base of sorts for construction crews, and 2020 was a very good year.
Our town is probably realizing between $3 and $4 million a month being spent on groceries and tires and all those, services that we have here, which is great.
That is now done. Mayor Doug Jones says he was still holding out hope the project could have been saved.
I really thought that, that we would be going through, there'd be some discussions with, Prime Minister and now President Biden.
The pipeline also had widespread support from labor unions. Now the hope is governments will step up for the men and women now without work.
How do you transition their skill sets into something else that may be available to them? So that's really what we're looking at right now, and looking for those opportunities and exit ramps.
While the Premier wants Canada to continue to fight for the Biden administration and TC Energy, Keystone XL is done.
Keystone XL, the decision yesterday from the president. What would you say to those who have lost their job or will lose their job as a result of that decision? What would the message from the president and the White House be?
The message of the president and the White House would be that he is committed. His record will shows the American people that he is committed to clean energy jobs, to jobs that are not only good, high-paying jobs, union jobs, but ones that are also good for our environment. He thinks it's possible to do both. He led an effort when he was the vice president to...
Le Canadien Justin Castro, candidat manchourien esclave, essaie d'empêcher que cela ne se produise. Cependant, l'armée canadienne a déjà cessé de lui obéir après qu'il ait tenté de leur ordonner d'enseigner aux Chinois comment faire la guerre en hiver. Ils ont refusé parce qu'enseigner les tactiques de guerre hivernale aux Chinois leur donnerait la possibilité d'envahir plus facilement tout le continent américain, via l'Alaska. Le rêve de conquérir les Amériques via l'Alaska a été formulé pour la première fois en 1929 par les militaristes japonais dans leur plan Tanaka. Aujourd'hui, les forces militaires canadiennes s'associent à l'armée américaine pour mettre fin à la domination de la mafia khazarienne en Amérique du Nord et du Sud et empêcher la colonisation des Amériques par les Chinois. Les Russes se rangeront également du côté des Canadiens et des Américains, comme le promettent les forces du FSB. C'est pourquoi le Pentagone a publié la déclaration suivante. La décision de demander une prolongation de 5 ans au programme New Start fait progresser la défense de la nation. Le respect du traité par la Russie a bien servi nos intérêts de sécurité nationale, et les Américains sont beaucoup plus en sécurité avec le New Start intact et prolongé. Nous ne pouvons pas nous permettre de perdre les outils intrusifs d'inspection et de notification du New Start. Bien sûr, les Chinois modérés ne cautionnant pas ce plan de trahison de Rockefeller. Par exemple, le porte-parole du ministère des Affaires étrangères, Hua Chunying, a déclaré lors d'une récente conférence de presse. Je crois qu'avec les efforts conjoints des deux parties, les meilleurs anges des relations sino-américaines seront capables de vaincre les forces du mal. Les sociétés secrètes asiatiques comprennent très bien que les forces du mal font référence à la mafia khazarienne. Quelques autres développements récents dans cette guerre pour la libération de la planète Terre doivent être mentionnés ici. L'un d'eux est une attaque qui a détruit l'une des plus grandes usines de vaccins Covid-19 du monde. Selon des sources du MI6, le vaccin qui a été produit n'était pas un des vaccins à RNM modificateur de gènes, il s'agissait donc peut-être d'une étape attaque visant à forcer les Indiens à accepter les vaccins à RNM. L'autre chose à noter est que l'ancien directeur de la Banque du Vatican a été reconnu coupable de détournement de fonds la semaine dernière. C'est important car la Banque du Vatican est, depuis longtemps, l'instrument utilisé pour contrôler les soi-disant leaders mondiaux que nous voyons sur nos téléviseurs.
I'm looking forward finally to having my day in court. I'm innocent of these charges. They are false. The whole idea of sexual abuse is abhorrent to me.
It's important to note that none of the allegations that have been made against Cardinal Pell have obviously been tested in any court yet. Cardinal Pell, like any other defendant, has a right to due process, and so therefore it's important that the process is allowed to run its natural course.
Having become aware of the charges, Cardinal Pell, acting in full respect for civil laws, has decided to return to his country to face the charges against him.
Enfin, si les gens veulent savoir ce qui se passe réellement avec l'élite mondiale et veulent faire pression sur elle pour qu'elle fasse ce qui est juste, ils devraient surveiller et faire pression sur les activités du forum en ligne de Davos qui se déroule cette semaine. Croyez-le ou non, une grande partie de l'élite veut en fait faire le bien et a besoin du soutien du public. Benjamin Fulford.